Hello everyone, welcome back to the Genomics Bootcamp. This video is a follow-up on a similar one that I have done some time ago when we presented open access datasets from livestock species. In this video, we will highlight open access human datasets. I start with acknowledging the source material and frankly, it is just from one website, from the Estonian Biocenter. So the website is shown here, and I will also link it in the description of this video. The people in the Estonian Biocenter are really awesome because what they do on this website is a really simple one, basically a list of papers and the links to datasets that could be used for research. Well, you might or might not remember that in a video on this channel, I was sort of complaining that I couldn't find open access human datasets to analyze. And just a day or two later, there was an email from my good colleague Arsen Dostev from the Russian Academy of Agricultural Sciences, and he was suggesting me exactly this website. So thank you, Arsen, again for your kind suggestion. This is a really useful resource. But then I thought also other people could benefit from this finding, and I thought I will make a video listing some of the datasets. Now I have to say that there is a lot of datasets on this website, so not all of them are listed in this video. What I decided to go for are the newest contributions, so the newest papers, and they are listed according to the alphabetical order of the first authors. Also, if you remain until end of the video, there is at the very end a really awesome data set that is, at least from my perspective, really out of the ordinary, really worthwhile to check out. But let's start at the beginning with the paper of Chaubey et al. Titled like Sugar in Milk, Reconstructing the Genetic History of the Parsi Population. Well, the Parsi people are a small group of people whose ancestors migrated to the Indian subcontinent from the region that is now a modern-day Iran. In general, this dataset is really a neat one, already in the PLINK binary PET file format, and it is from 43 people from India and Pakistan with almost 300,000 SNPs. The next paper is from Scientific Reports, Eichstaedt et al. 2017, evidence of early stage selection on these two particular genes in Andean high altitude populations. So we have a very neat aspect of the high altitude here in this paper. So the aim of this paper was to identify genetic variants that harbor signatures of recent positive selection that may facilitate physiological adaptations to hypobaric hypoxia. Also, in this case, we speak about PLINK binary datasets from 12 people and more than 1.2 million SNPs. Also, there is a README file which you open and then you find even more sources and even more datasets there. The next paper holds a little bit different data. Ilumäe et al. 2016 published in the American Journal of Human Genetics, the human Y chromosome haplogroup N. This dataset contains chromosome Y captured data and sequences from 43 samples. The target enrichment design was utilizing 67,000 capture probes for sequencing at least 10 megabases on the Ilumäe platform with more than 60x coverage. What you find in these datasets are VCF files for 11 individuals and .fastq files for 32 samples. The paper of Järve et al. 2019 is also really interesting. It is titled Shifts in the Genetic Landscape of the Western Eurasian Steppe Associated with the Beginning and End of the Scythian Dominance. What you find in this dataset is PLINK binary PET files from 31 people, and these are these ancient samples of Scythians who were actually ancient nomadic people of Eurasia. Each sample is genotyped for more than 500,000 SNPs. The study of Mörseburg et al. 2016 was published in the European Journal of Human Genetics, titled Multi-Layered Population Structure in Island Southeast Asians. Well, this paper analyzes a lot of individuals from Southeast Asia and also some new genome-wide genotype data from various countries. What I found in the attached PLINK binary PET files were genotypes from 139 people, genotyped for more than 700,000 SNPs. The genomic impact of European colonization of the Americas was studied by Ongaro et al. 2019, published in Current Biology. It deals with the complexity of the admixture dynamics that shaped American populations and also evaluates the dramatic impact of events after the colonial era. The PLINK binary PET files contain genotypes from 88 individuals for more than 1.2 million SNPs. Pagani et al. 2016 published a paper in Nature titled Genomic Analysis Inform on Migration Events During the Peopling of Eurasia. Here I have to say that peopling is a very strange word for me. Anyway, the dataset contains genotypes of 402 individuals, both in PLINK files and VCF files that are packed in two large zip files, both of them about 1.5 gigabytes after unpacking. If you go to the mentioned webpage on the Estonian Biocenter, you find links to even more data sources related to this paper. Also, in this particular case, there is also a link to a YouTube channel that discusses this data. The Indian populations were studied by Pathak et al. 2018, who published a study, The Genetic Ancestry of Modern Indus Valley Populations from Northwest India, published in the American Journal of Human Genetics. The study contains 45 new samples from 4 populations from India. It is in the PLINK binary PET format, genotype for more than 700,000 SNPs. The following paper is from Saag et al. 2017 and is titled Extensive Farming in Estonia Started Through a Sex-Biased Migration from the Steppe. It deals with the data extracted from DNA of skeletal remains from 10 individuals from Estonia dating 4.5 to 6.3 thousand years ago. It is actually a sequence data in BAM files, which are about 1 gigabyte each. In a README file, you will find additional description on the data processing that was given by the authors. Saag et al. 2019 discusses the arrival of Siberian ancestry connecting the Eastern Baltic to Uralic speakers further east. Again, it is DNA samples from quite ancient finds extracted from the teeth of 33 individuals from the Eastern Baltic dating to 3,200 to 400 years ago. It is in a format of binary PLINK files, so very convenient to use, genotypes of 56 people. Here I have to say that there are actually 2 data sets and they appear to be the genotypes of the same 56 people. First dataset contains almost 1.2 million SNPs and the second dataset about 500,000 SNPs. What I didn't check if these 2 SNP sets are overlapping or they are distinct from each other. So this is for you to find out. And one more paper from Saag et al., this time from 2021. So it's a very recent one. Genetic ancestry changes in Stone to Bronze Age transition in the East European plain, published in the Scientific Advances journal. This paper reports new genome-wide sequence data from 30 individuals from the understudied western part of the present-day Russia, including 3 Stone Age hunter-gatherers and 26 Bronze Age farmers from the corded ware complex Fatyanovo culture. The genotypes from 47 individuals are in PLINK binary PET file format, and again, a lot of SNPs, so more than 1.2 million SNP per individual. And the last conventional dataset presented in this video is from Saupe et al. 2021, published in Current Biology, and the title of the paper is ancient genomes reveal structural shifts after the arrival of the steppe-related ancestry in the Italian peninsula. Using this data, the authors explored the genetic history of the Italian peninsula through the Chalcolithic and Bronze Age transition. This data is available as PLINK files from 22 individuals and again about 1.2 million SNPs per individual. And this last paper in this video is the promised curiosity, at least for me. The paper is titled Inferring Patterns of Folktale Diffusion Using Genomic Data. So it puts somehow together the folktales, the geographic data, and genetic distances. It identifies 15 tales that are more likely to be predominantly transmitted and locate the putative focal areas for a set of tales that are spread worldwide. It is not a conventional data set. What I understand is this space mix software or approach, and the data set is actually the input files for this space mix approach, which is actually a method for inferring and visualizing spatial patterns of population genetic structure and admixture. So what you get are coordinates, the matrix of the 586 fairy tales, and the genotypes of the human groups. I like this connection of fairy tales and genomics very much, and therefore I decided to highlight this paper all among the others. Of course, the other papers are also really important and interesting, giving us open access data. Many times this data is in the very convenient PLINK binary ped format, but also VCF files or other sequence-related files, which could be very useful in our further and future research. If you like this video, make sure to leave a like, and also if you want to see similar videos in the future on this channel, leave a comment below and perhaps tell what kind of data you are interested in. For today, I thank you for your time and have a very nice rest of the day.